Hello, welcome. My name is Dr. Celeste Stewart. I'm the Patient Engagement Manager for the National Ataxia Foundation, and it's my pleasure to welcome you to this session all about gluten ataxia. It is my privilege to introduce today's speaker, Dr. Marios Hadjivassiliou, a consultant neurologist and academic director of the Department of Neurosciences, Sheffield Teaching Hospital, NHS Trust, and the University of Sheffield. His primary research is the neurological manifestations of gluten-related disorders, and the focus of his MD thesis and subsequent fertile area of research over the last 30 years. His pioneering research resulted in defining previously unrecognized disease entities such as gluten ataxia, gluten encephalopathy, and gluten neuropathy. He has published extensively in high-impact journals, and he runs a weekly gluten sensitivity neurology and ataxia clinic, and receives referrals from all over the United Kingdom and internationally. And with that, Dr. Hadjivassiliou, the floor is yours.
Thank you very much for the introduction. Thank you all for joining. And also thank you for the invitation to talk about gluten ataxia. I thought you might be interested in a little bit of history, starting with why is celiac disease spelled differently in Europe and in the States. OE. Well, celiac disease, which most of you will be familiar with, was first described by a Greek doctor called Aristeos from Cappadocia in ancient Greece on the 2nd century AD. And his books were first written in Latin. And you see that the Greek spelling of kiliakaki, which is celiac disease, and kilia means abdomen, is spelled with E, which is OI, and therefore the Latin version of that was OE, hence why it's spelled like that. But North America always likes to simplify things, which is why they got rid of the O. Anyway, then there was a big gap following the description by the ancient Greek doctor until 1888, when Samuel G., who was an English pediatrician, described the modern, if you like, clinical picture of celiac disease. But believe it or not, it wasn't for another 50 years plus before the Dutch pediatrician Willem Dicke discovered the connection between celiac disease and wheat. And it was a stroke of luck, really, because during the Dutch famine in 1944-45, during the Second World War, Willem Dicke was looking after children with celiac disease, and because of the lack of bread, they all got better. And then, of course, when the supplies increased, then he noticed that the children were getting sick again, and that helped him make a connection with wheat as being the cause of celiac disease. Of course, celiac disease no longer looks like this, and is not any longer a disease of children. By the way, the little girl on the right-hand side is the first girl to be treated in the UK after the publication of the work by Willem Dicke. And you see that the disease was really quite severe, with malnourishment because of malabsorption, lack of albumin, and therefore adults would end up with dependent edema and so forth. Of course, we now know that celiac disease, if you do a prevalence study, affects 1% of what we call healthy population. And then the issue as to whether or not other parts of the body, in fact, is not new. The extraintestinal manifestations were firstly highlighted by this paper that demonstrated the presence of a skin condition known as dermatitis herpetiformis. And the authors of this paper back in Lancet in 1966 showed that patients with dermatitis herpetiformis had an inflammation of the bowel that was identical to what is seen in celiac disease, but that if you went onto a gluten-free diet, the skin rash disappeared. And some of these patients actually did not have any evidence of inflammation of the bowel. This is what dermatitis herpetiformis looks like. It's a itchy vesicular rash, usually over the extensor surfaces, so knees, elbows, back, and so forth. And it gets better when patients go on a strict gluten-free diet. The other important issue that is worth bearing in mind is that gluten sensitivity without enteropathy, without celiac disease, is actually not a new concept. Mike Marsh, who was a gastroenterologist in Manchester, he died a couple of years ago, did some really elegant work demonstrating that depending on the amount of gluten that you sensitive, the typical celiac abnormality that you see in the bowel. But obviously, some patients may have a normal looking bowel and still be gluten sensitive. And hence, the nomenclature nowadays, when people have a biopsy to look for celiac disease, uses the so-called Marsh classification. So it's best if you think of the whole group of diseases as gluten-related disorders. So it's a group of autoimmune diseases triggered by eating gluten. And gluten, remember, is found in wheat, barley, and rye, which includes celiac disease. We know a lot about celiac disease, the skin condition called dermatitis herpetiformis, and of course the area of our interest today, which is the gluten sensitivity related neurological disorders, and in particularly gluten ataxia. So when we refer to gluten ataxia, we are talking about patients that may or may not have celiac disease, but all of them have in their blood evidence of antibodies that signify that they are sensitive to gluten. And either they present with or develop neurological problems. So how did this whole journey start for us? Well, back in 1994-95, we considered the possibility that gluten sensitivity may be playing a part in some neurological conditions. So we did this study that was published in The Lancet in '96. And basically, what we did is use the available markers at the time, anti-gluten antibodies, and we just screened patients with neurological dysfunction of unknown cause. And a lot of these patients were patients with ataxia. And we showed that there was a very high prevalence of these antibodies when you compared it to healthy blood donors. And in fact, when we went on to do biopsies in these patients, we found that the prevalence of celiac disease in this group of patients was 16%, which is 16 times more than in the healthy population. And that's what kind of triggered this interest in the neurological manifestations of gluten sensitivity. Now, there was one other thing to consider from that paper, which is that the majority of these patients who had the antibodies actually didn't have celiac disease. And yet, when they went on to the gluten-free diet, they got better. So we introduced the concept of gluten-related neurological dysfunction without celiac disease. And this is where we are now. So as Celeste said, we run a weekly gluten sensitivity neurology and of course ataxia clinics, and we've got a lot of patients. The last count is 2050 or so, but is to show you that the most common manifestation of this condition is ataxia. But only about 26% of these patients will actually have enteropathy once you've done the biopsy. So let's talk a bit about gluten ataxia. So we went on, after we collected a few cases, to provide clinical imaging, neurophysiological, and some postmortem material to characterize gluten ataxia. And we in this paper published in the Lancet a couple of years after the previous one, we introduced the term gluten ataxia, which we thought was suitable for this type of ataxia. So how do we make a diagnosis in the first instance? Well, this is where diagnosis can be a bit problematic. Neurological testing, blood testing, and there are antibodies that are very sensitive and very specific to the presence of an enteropathy, which what we call celiac disease. And if you go to a hospital and they want to test you for celiac disease, one of those two antibodies is what you will have done, either endomysium antibody, EMA, or tissue transglutaminase, also known as TG2. However, as I said to you before, only 28% of patients with gluten-related neurological dysfunction have celiac disease. So if you only use these antibodies, you will miss the majority of patients who have a potentially treatable neurological problem. So anti-gliadin antibodies, which is what we use, are sensitive to the whole spectrum of gluten-related diseases, but unfortunately, they are not longer readily available in many of the NHS labs and in some of the other countries. And of course, TG6 antibodies, which we'll come to in a little bit later, are more sensitive for the neurological manifestations of gluten sensitivity. So a patient comes, you do your blood test, this is what you get from the lab, and this is the classic celiac disease patient. And you can see that the endomysium antibody is positive, the tissue transglutaminase antibody is very high. So when we get a referral, for example, for a patient with a progressive ataxia, often the referring neurologist will have already done the tissue transglutaminase antibodies to look for celiac disease, but these are negative. So they want us to see this patient and decide what else can we do to check for causes of ataxia, because we are an ataxia center, a National Ataxia Centre as well. So of course, when we do the antibody testing, you see that indeed they don't have celiac disease because the endomysium and the tissue transglutaminase antibodies are negative, but they are positive for gliadin antibodies. So this patient has gluten ataxia without celiac disease. And the other pitfall in the diagnosis would be somebody like this, somebody who's got celiac disease, which they had for a long time. They admit that they've not been brilliant with their diet, but they are now back on track and they are very careful with their diet, yet they seem to be developing ataxia. So what's going on here? When they come and they have the test, you can see that they are sufficiently gluten-free to normalize the antibodies that tell you about celiac disease, which is the endomysium and transglutaminase, but they're still positive for gliadin antibodies, which means that they eliminate all the antibodies. And then what do? We get them seen by our expert dietitian, who gives them further advice about cross-contamination and other issues of what they're eating. And you can see that this patient then is able to eliminate all the antibodies and improve their neurological problem. How common is gluten ataxia? Well, it is the commonest ataxia you will ever encounter. Here is based on 3,000 patients that we look after here in Sheffield. And the big blue section is gluten ataxia. And the second biggest is the genetic types of ataxia that I'm sure some will be familiar with. What are the clinical characteristics? I think what you need to take out of this slide is that everybody with gluten ataxia will have problems with their gait. And this is because, like all of the immune ataxias, they primarily affect the middle part of the balance center, as we call it, the vermis, which is responsible for walking. All of them have abnormalities within the vermis, and all of them have problems with the gait ataxia. Now, There are other phenotypes that we can encounter in gluten ataxia, which are fortunately more rare, but it's important to be aware of. And I'll share with you some. The first phenotype, which you can only see in immune ataxias, you never see this in genetic ataxias, is this rapidly progressive ataxia. And we put together a few case series of patients with gluten ataxia just to highlight this problem. And I'll show you a video of this example. Now, of course, when you are faced with somebody who is rapidly progressing, what you need to do is try and intervene above this so-called threshold line, because at that stage you are dealing with a restorable stage of disease. If you pass that threshold, you no longer are going to be able to restore the so-called cerebellar reserve, and you're going to be left with permanent ataxia. So let me share this video with you.
Big circle, okay, and I do the same with this leg.
So this is a young patient, as you can see.
Right. British Constitution. Okay. Nose, touch my finger.
So the treating neurologist was so concerned about the rapidity of progression that rang me and asked if we could see the patient as an inpatient, which is what we did.
Close your eyes.
This is when the video was taken.
That's great. Okay, so left and right. That's it.
So we did the appropriate blood test. We checked to ensure that the patient didn't have any cancer, which often can cause this rapidly progressive ataxia. And we found him to be positive for TG6 antibodies, so he's gluten sensitive.
Are you able to stand with your feet any closer together? And close your eyes. Okay. I'll just take a few steps this way. Up.
And this is the same patient a year later on a strict gluten-free diet.
Tell us, hello, my name is Matalik Cowboy. I live in Stafford. I'm 20 years old. And how much wood could a woodchuck chuck if a woodchuck could chuck wood? What do you think about how I walk?
All right, you don't need to do a catwalk.
So why did he do so well? Because he was diagnosed early, because he's young, and he went on to the gluten-free diet and stuck to it, and therefore very good outcome. The other phenotype that is fortunately rare, but very easily recognizable, is what we call myoclonic ataxia, sometimes referred to as the central nervous system hyperexcitability spectrum. Why is this very important? Because it's often associated with something called refractory celiac disease. Refractory celiac disease means that you pass the stage where avoiding gluten is sufficient to normalize things, and often you require immunosuppression to help that recovery process, or sometimes it can be neuropathy associated lymphoma, which is really very serious and very difficult to treat. So this is a compilation of films from patients with this condition. So it can affect different parts of the body. Here is the arm twitching. Here is again the arm. Patients have what we call stimulus sensitive malclonus, which makes it even more difficult for them to do certain movements. So it's very, very disabling.
It's difficult for you, but you gotta go.
Patient is trying to put his foot on the pedal, trying to get off the floor, but every time he does that, it's bouncing back. And here you see speech arrest. Again, as a result of this malclonus. Now, those two phenotypes are rare, you'll be pleased to know, but it's good to be able to recognize them for us because we need to intervene very, very quickly. So how do we treat and how do we monitor patients with gluten ataxia? So we did a study published many years ago in which we showed that there was improvement of the ataxia within a year of strict gluten-free diet, even in those patients who didn't have celiac disease, just had the antibodies. And of course, the monitoring can be a blood test showing that these antibodies are eliminated, as you can see here. But even better is a MRI scan of the brain that includes something called spectroscopy. And I'll just want to spend a bit of time to explain to you what we mean by that. So when you do the MRI scan, you can actually use some software that puts a little rectangle on the bit you are interested in. In this case, is the cerebella vermis, the middle part of the balance center. And that allows you to do spectroscopy, and the spectroscopy comes up with these peaks. The first peak you see is called N-acetyl aspartate, and is a metabolite that it's of healthy functioning cells. So the higher this is, the better. The second metabolite is creatine, which doesn't change in health and disease. So when we take the ratio of Na to creatine, we come up with a numerical value that normally should be well above 1 if you are normal. As you can see here, is below 1, and that's because this patient has gluten ataxia. But when they go on the diet, you can see that the NAA increases because the cells are functioning much more healthily, and therefore this is a very, very useful monitoring tool over time. And this is what we use in Sheffield. So here is each line represents a patient, and here we have the ratio of Na to creatine, and you can see that everybody improves on a strict gluten-free diet. Here is a small number of patients who did not go on the diet, and you can see that that ratio is decreasing, which means that they are getting worse. And finally, there is a third group of patients who are on the diet, but they are not very strict with the diet. And you can see that there is some decline here, but it's not as steep as those who are not on the diet. If you put all these things together into 3 main lines, you can show that those who go on a gluten-free diet and are strict with it improve. Those who don't go on the diet get worse, and those who are on a partial diet get worse, but slowly. And this graph, you will agree, is identical to the previous one, but you see at the title here that this represents all the patients who have celiac disease, and the next represents those who don't have celiac disease, but the outcome is identical. So why is it so important to make an early diagnosis and treat? Well, because the whole process of damage to the cerebellum starts from early, and therefore the sooner you intervene and put patients on a gluten-free diet, the more likely it is to preserve the cerebellar reserve. How common is it? Well, here you are. This is a study we did funded by Celiac UK, where we examined and did brain imaging in every patient that was diagnosed with celiac disease in the classic pathway, the gastrointestinal kind of problems presenting to the gastroenterologist. So we had 100 patients, all of which had scan, were examined by neurologists, and to our surprise, 46% of these patients actually had abnormal spectroscopy of the balance center at diagnosis. So already the process of damaging the balance center was there. And in addition, a lot of these patients, or 19% to be precise, had these so-called white matter lesions over and above what you'd expect for their age. And this is what we tend to see in an entity called gluten encephalopathy. Now, I'm talking about this sort of degree of mild ataxia when you actually examine the patients, and this is one of those patients with celiac disease, and get them to do some simple tasks like you see here, standing on each leg in turn and tandem walking. They are actually not quite normal for somebody who's only 19 years old. You would expect them to do this without any difficulty, whereas they're beginning to struggle a bit. And if you do the spectroscopy of the balance center, you see that it is abnormal. So what you need to do is intervene at this stage on the left-hand side before the cerebellum ends up being very, very atrophic, shrunken, at which stage you're not going to be able to reverse the process. And just for contrast, to compare with that girl I showed you, here is a patient with gluten ataxia who we diagnosed 20 years after he developed his ataxia.
Can I ask your name?
William Pearson.
And your address?
Addressing Lagos Grove. The home in your below Lagos Grove.
Thank you very much.
Whitford, Cheshire.
Fine. Could I ask you, Mr. Pearson, to put your arms in front of you? Okay. And close your eyes. And open your eyes. Just relax this arm. Let it go down. Just relax it. Okay. I know it's difficult for you. Could you just try with this finger to touch your nose gently?
Very disabling.
Touch my finger now.
And
that's fine.
All you can achieve at this stage is, of course, stabilization rather than necessarily improvement. So why sometimes patients may come back and say, well, actually, I've not noticed any improvement. By far, the commonest cause is the poor adherence to a strict gluten-free diet. And you can show that by repeating the antibody testing. You want the antibodies to become negative. It could be that the patient is already very disabled, as I showed you in the previous video clip. And in that situation, the spectroscopy can be helpful because you can show that the spectroscopy is improving. It shows you that the diet is having an effect, even if that effect is not clinically apparent. Obviously, you need to consider the possibility of refractory celiac disease, but those cases are very, very rare. And then finally, you need to review the diagnosis. You may have got it wrong. You may have somebody who has a genetic ataxia, and hence why that's continuing to deteriorate despite the gluten-free diet. But I work at the Sheffield Ataxia Centre. We have access to whole genome sequencing. We do work up these patients extremely well, and we are pretty confident that that's the correct diagnosis, and we have excluded everything else. We did a follow-up study following that original study I mentioned to you, patients with newly diagnosed celiac disease. And it's just to demonstrate the importance of the monitoring, because this was 7 years after the original study. And we were able to show that these patients whose antibodies were consistently positive, even at 7 years down the line, they had more cerebellar gray matter atrophy than those who were negative. And therefore, those who don't adhere to a strict gluten-free diet and they are constantly positive, they are at risk of developing ataxia later on in life. So a little bit about the pathophysiology to finish off. and I have to introduce this story, which I think is quite an interesting story. And this is regarding an enzyme called transglutaminase. So these are enzymes that are found in humans and other species that are involved in tissue repair. So if you cut your skin and have blood, the transglutaminase will bind proteins together to try and stop the bleeding. So and at the same time, they are very good at breaking down gluten into small peptides. And unfortunately, these are the peptides that then cause the immune response for those who are gluten sensitive. And therefore, you end up with your celiac disease and your ataxia and so forth. Now, a big breakthrough came in 1997 when tissue transglutaminase, also known as TG2, was found to be the autoantigen in celiac disease. And it kind of proved that celiac disease was an autoimmune condition. A couple of years later, or 5 years later, the TG3, which is an epidermal transglutaminase, was shown to be the autoantigen in dermatitis herpetiformis, the skin version of celiac disease that I showed you earlier. And at the International Celiac Disease Symposium in Paris in 2002, I met a scientist called Daniel Eschliman, who was working in and still works in Cardiff University in Wales. And he presented a new discovery of another transglutaminase called TG6, which was primarily expressed in brain. And this is the developing rat cerebellum. The black areas is where transglutaminase 6 is expressed. So I met Daniel at that meeting and said, could it be that TG6 is the target for those patients where the immune response is targeting the brain rather than the skin or the gut? And we worked together for 6 years at the time, and were able to publish this paper where we showed that autoantibodies in gluten ataxia recognized a novel neuronal transglutaminase, which of course was TG6. And then colleagues in Trieste we collaborated with had experience in using mouse models in ataxia. So you get the mouse to stay on the rotarod as a means of demonstrating how good their balance is. And when you inject serum from patients with gluten ataxia in these mice, you can show that they develop ataxia based on the rotarod test. If you sacrifice the animal, you can then show that there's a lot of staining in the cerebellum, so the antibodies in patients with gluten ataxia are immediately targeting the balance center. And the researcher from Trieste then came to Sheffield and did some more experiments just to sort of try and see what the differences were between transglutaminase 2 and transglutaminase 6. And obviously, as you might expect, 6 seems to recognize much more Purkinje cells, which are important in cerebellar functioning and deep cerebellar nuclei, whereas TG2 is primarily around brain vessels. Going back now to the publication of our paper about the 100 patients with celiac disease, 40 of those 100 patients had TG6 antibodies. The others did not. So we were curious to know what the difference was between the 40 who had the antibodies and those who didn't. And when we looked at their brain imaging, we were able to show that the gray matter in the balance center, the cerebellum, was less in those who were positive for TG6 when compared to the remaining group. In other words, if you have a TG6 in your blood when you have celiac disease, it tells you that you are more prone to neurological. And these are some experiments. Don't bother with the details. I'm just going to very briefly tell you what they are about, done by Mara Floare, one of our PhD students who's working in the lab. So basically, what we were able to do is demonstrate that in the spinal fluid of patients with gluten ataxia contains TG6 antibodies. And similarly, you can actually take away the TG6 antibodies from the spinal fluid by using a methodology that picks up all the TG6 antibodies. And if you use serum from patients with gluten ataxia, you can show that, again, it reacts strongly with Purkinje cells. And again, if you remove the TG6, then you can show that you reduce that staining. So there's an interaction between TG6 antibodies and cerebellar tissue, and hence why we're so insistent in demonstrating the elimination of the TG6 antibodies to allow the full recovery of patients with gluten ataxia. We did gut biopsies as well in these patients with gluten ataxia who don't actually have celiac disease. And interestingly, you can see in the gut that they have plasma cells. Plasma cells are the cells that produce antibodies that are producing TG6 in the gut, which of course is the place where your body first comes in contact with gluten. And one other important finding made in China was that identification that a mutation, a faulty gene in the gene that expresses TGM6, which is the protein that is associated with the TG6 antibodies, causes spinocerebellar degeneration known as SCA35. So what that tells us is that there's something about the TG6 protein that is important for cerebellar functioning. Hence, it's not surprising that having these antibodies results in cerebellar dysfunction. So our experience with TG6 antibodies originally was based on the in-house assay produced by Daniel Eschliman in Cardiff. And I already told you that 40% of patients with celiac disease in the UK have TG6 antibodies. But interestingly, when we tested a cohort of patients, samples from Finland, only 14% had TG6 antibodies. And that, I guess, tells us that there might be some geographical variations. So some populations might be more prone to developing neurological problems rather than others. We also looked at the pediatric cohort from Trieste, Italy, and we found it in a quarter of those children having TG6 antibodies. And we also looked at another Finnish population of patients with dermatitis herpetiformis, and this time the prevalence was much higher. And in healthy control, it was about 4%. We then introduced the TG6 antibodies into our immunology lab here on a commercial, or on an NHS basis, based on a commercial assay. And the introduction was in 2019. And that was a big step forward because it meant you can use them on an everyday clinical practice. So every patient who comes with ataxia to Sheffield will have this done. And this is our data. Firstly, reassuringly, all those patients who we define as having gluten ataxia based on the fact that they had positive GLADin antibodies, 96% of those were positive for TG6 as well. So that's a nice correlation between the GLADin and the TG6 antibodies. We found them to be present in 50% of patients with the classic celiac disease, very similar to the 40% that we found with the in-house assay. But very interestingly, we found that in a group of patients with idiopathic sporadic ataxia, in other words, no other cause found, and negative for all the other antibodies that we do for celiac and gluten sensitivity, 77% were positive for TG6 antibodies. And this is a kind of lab picture. So negative for endomysium, negative for transglutaminase, negative for GLADin antibodies, positive for TG6 antibodies. And these patients improve on a strict gluten-free diet the same way that any patient with gluten ataxia does. And because we also had data on these patients before we were able to make a diagnosis of gluten ataxia when the TG6 became available, you can see here how their spectroscopy was deteriorating, and then they go on the diet, and then it's improving. So TG6 antibodies can be sometimes the only marker of gluten ataxia. So what's currently in research? Well, we are currently have some funding from Celia UK, Ataxia UK, and the Sheffield Hospital Charity to try and improve the diagnosis and management of gluten ataxia by getting patients from other ataxia centers and other neurologists being referred to us, and it's a clinical referral, and we're therefore able to screen these patients for gluten antibodies and TG6 antibodies. And if we make a positive diagnosis, then they go on the gluten-free diet, and we repeat the scan a year down the line. What else is of interest? Well, one important question we would like to address is the use of immunosuppression to aid and speed recovery, at least at the early stages of the diagnosis. The diet and the elimination of the antibodies takes about a year to manifest, and therefore during that year you might continue to get worse before you get better. And therefore, is there a mileage in using drugs that suppress the immune system to prevent that further damage to your balance center before the diet has its full effect? And along those lines, there's a paper that hopefully will be out very, very soon by one of our researchers, Ian Kroll, that shows that if you have persistingly positive TG6 antibodies, then that is associated with a worse neurological outcome. Along the laboratory work done by Mara Flores, we have produced some cell lines from human skin fibroblasts from patients who have SCAR35, and you can reprogram these cells into astrocytes, which are neurological cells. And then it allows you to see what exactly is the problem with those cells that have these mutations, and how does TG6 influence cerebellar function and dysfunction. And this is just to show to you how long it can take for these antibodies to normalize. So this is just one example from a patient. Look at the dates: January 2020, positive TG6 antibody, and it's not until September 24 when the antibodies become negative and then eventually completely disappear. So during this time, you could argue that there's still some damage being done, and therefore, if you were to use immunosuppression at these early stages, maybe that would be an additional benefit to the patient. So think of gluten ataxia as an iceberg. I think that's the best characterization, where on the surface you have your classical celiac disease, and some of these patients may develop ataxia because they are not very good with their diet, for example. Below the surface, you have those patients with celiac disease who present just with ataxia. And fortunately, you would think, at least in the UK, that neurologists will be thinking about celiac disease when they see a patient presenting to them with ataxia. But even deeper in the ocean, and the bigger part of this iceberg, is these patients who have gluten sensitivity. They don't have celiac disease. They might just be positive for anti-gluten and/or TG6 antibodies, who present with ataxia. And if those tests are not applied in this cohort, these patients will continue to follow a progressive course. And finally, a quote by J.M. Case, who is a very famous person, economist actually, who said that the difficulty lies not so much in developing new ideas as in escaping from old ones. And I think this applies very much to our work here. So I wanted to thank you for taking the time to listen to my talk, and I'm more than happy to answer any questions that you have.
Thank you so much, Dr. Hadjivassiliou. We're going to dive right into questions. As a reminder for attendees, if you have questions, please put them in the Q&A window at the bottom of your screen. Our first question from an attendee: is it common for people with gluten ataxia to have vitamin B12 deficiency?
I wouldn't say it's common, but we always look for it, because if particularly patients who have celiac disease are more prone to malabsorption.
That's good to know. We have another attendee who asks, can any neurologist diagnose gluten ataxia, or do you need to see a movement disorder neurologist?
I don't know how it works in the States. Here, we have ataxia centers. I think you have in the States as well. So
We do.
You can be referred to the ataxia center. If it's far from where you are, then your local neurologist, some of the ataxias get seen in the movement disorder clinics, I believe. And accessibility to those tests is the problem. So most immunology labs in any decent-sized hospital will be able to test for celiac disease, but they won't be able to test for gliadin and TG6, and that's where the problem lies.
And then a related question from an attendee: are you familiar with any doctors in the United States who specialize in gluten ataxia or celiac disease and related conditions like you do?
I always get asked that question. I'm afraid not. I mean, I know some of the ataxologists in the States. I think, fortunately or unfortunately, a lot of my colleagues focus a lot on genetic ataxias rather than immune ataxias, which gluten is one of. I know people who have a lot of knowledge in celiac disease, and they're very open to look at gluten sensitivity. At Columbia University, Professor Peter Green, for example, is somebody who's extremely experienced in celiac disease, and sometimes I mention to patients about him. But we just need to increase the awareness and also increase the interest in gluten ataxia, I believe.
And then moving on to our next question. This is from an attendee whose question refers to the pool of 100 patients, of whom 46 showed brain impact on the scans. For this pool of 100 newly diagnosed celiac disease patients, were these all new celiac disease patients, or were these individuals in the pool because there was a suspicion of ataxia already?
No, they had no suspicion of ataxia, nothing at all. There were patients presenting to the consultant gastroenterologist in the usual pathway where celiac disease gets diagnosed. So they might have got anemia or bowel symptoms and so forth. And that's why it's so surprising, because in fact, what was also interesting is if you ask the patients, do you have any problems with your balance? There was a huge number, more than 50%, that said yes. Not sufficient for them to seek advice specifically for that problem. And of course, they wouldn't necessarily discuss it with the gastroenterologist. And as I showed you in that video with the young girl, if you get them to try and tandem walk, they were struggling. And that reflects the fact that their brain scan is abnormal. But those are the lucky ones, because they get diagnosed and they go on the strict gluten-free diet and they do very well. And if you look at the age at diagnosis of the classic celiac disease, is about 43 for the adults. If you look at the age of diagnosis of celiac disease, if you present with ataxia, is 53. And therefore, you can imagine that somebody progresses from 43 to 53, and then they have a full-blown ataxia.
That makes sense. Speaking of the strictness of a gluten-free diet, we've had a few people ask a very similar question around how much gluten must one consume before antibodies register. Or I've heard that even just a few bites of gluten constitute not an adherence to a gluten-free diet. Is this right? Could you talk a little bit about the strictness of the diet and how much gluten you need to consume before you are seeing these antibody signals?
The truth of the matter is that what we encourage people is to be 100% gluten-free. And it's a tricky situation here, because of course, if you are in doubt, you are at the mercy of the restaurants and the chefs and whatever. If you are eating at home, you can be a bit more careful. We talk about cross-contamination. What we mean by that is that you, for example, use a different toaster to somebody who uses ordinary bread. You use a different knife to butter the bread, the gluten-free bread. You use a different chopping board. And sometimes those issues can be sufficient to make a difference between a positive serology and a negative serology. And as you saw with some of the examples that I mentioned, you can be sufficiently strict to normalize your celiac antibodies, but your uncladins and your TG6 can still be positive. And our experience with TG6 is that it's probably the last antibody to go and probably the most sensitive. So, and then you have this other issue of the food being labeled as gluten-free when might contain a small amount of gluten beyond a level that is allowed to be called gluten, made out of barley and so forth. And my advice to patients is that just don't go for gluten-free wheat or gluten-free barley or rye. Just people use different flours from different pulses to produce bread and so forth, and that's probably the safest thing to do. There is one issue that I get questioned a lot about oats. Oats are not do not cause normally a reaction to in the context of gluten sensitivity because they are completely different proteins. The main problem with oats has been the cross-contamination, the fact that they're produced in areas where wheat is often produced. So you can get gluten-free oats, and those should be fine for the majority of patients. There is a very, very small number of patients who seem to also react to oat proteins. And in that situation, it becomes obvious because when you withdraw oats, then the antibodies disappear.
Thanks for clarifying that.
What you really need is a very experienced dietitian, and that's what we have here, that know all the ins and outs. Because at the end of the day, they are the most important people in the monitoring process.
It sounds quite complex, that when you're learning for the first time, but I'm sure once you get into the hang of it, things become a lot more normalized and you get into the habit of eating with these different sets of rules.
That's right. And also there's a lot of useful information in the web of the Celiac UK, and I'm sure it must be the same with the equivalent organization in the States, the Celiac organization.
I also will take this opportunity. We recently hosted a new webinar series that the National Ataxia Foundation is putting on, Cooking with Troy, who is a member of our community who has gluten ataxia, and all of his meals that he makes are gluten-free, and also shows you different tips and tricks for cooking when you have low dexterity. But that's available on our YouTube page. We'll move on to the next question. This is also relating to diet and symptoms and that connection there. How quickly after starting a strict gluten-free diet do people have improvement in their symptoms? How long do you typically monitor to see if there might be any benefit from a gluten-free diet versus considering another cause of ataxia?
Good question. So the gastrointestinal symptoms, if you have them, and of course the majority of the neurology patients will not, improve very quickly. When I say quickly, within weeks, okay? We haven't talked about gluten encephalopathy, the so-called foggy brain, headaches, and so forth. Those kind of things also tend to improve quickly. The ataxia takes a year. So what we tend to do is see the patient in about 6 months after they've seen the dietitian, given the advice, see them at 6 months, repeat the antibody test, and at least show that the antibodies are going down. They may not have gone completely at that stage, but the repeat brain imaging we tend to do at 1 year, and that is when it shows that there is usually significant improvement in the spectroscopy measurements, and that's when you begin to see the patient noticing the clinical improvement. So it takes time. As regards the issue of when do you start thinking about could it be something else, usually after a year. And of course, you start thinking about something else if the antibodies, despite the fact that the antibodies have disappeared, the patient and the scanning suggests that things are getting worse. That's unlikely, or what you see with gluten ataxia. And of course, like I said to you before, we would have already looked at other possibilities. In the majority of our patients, we would have done whole genome sequencing, looked at all the genetic causes and so forth. So most of our patients would have had everything else ruled out. Now, what happens then if you have a patient that you are happy that they have gluten ataxia, they've eliminated the antibodies, but they seem to be still following a progressive course, having ruled out everything else? That's when we use immunosuppression. So you will use medication. We tend to favor mycophenolate, which is one of the drugs that suppress the immune system. And at that stage, we will introduce it as a means of providing more stability and allowing the immune system to settle with a little bit of help with the medication.
And then we have time for one last question. We have a few attendees who have genetic forms of ataxia, and they are wondering if there's any known connection between genetic forms of ataxia and gluten ataxia, or if a gluten-free diet might be beneficial for them, even if they don't have gluten ataxia specifically, they have another diagnosed form.
Excellent question. So inevitably, given that the prevalence of gluten sensitivity and celiac disease is quite high, you will have patients who have a genetic ataxia who are also gluten sensitive. And the question is, would that benefit their ataxia? And it's very interesting because if I come across a patient in that scenario, what I usually say is that, listen, you are gluten sensitive. The driver for your ataxia is the genetic cause. To try the diet, then there's no reason why not, and there might be some benefits. And of course, if the patient already has a lot of gastrointestinal symptoms or other symptoms that I would attribute to gluten sensitivity, of course, those will improve. What you see in terms of the ataxia, if you do repeat scans, is interesting. What you tend to see is that there is an initial improvement in the spectroscopy, but it's not sustained. And I guess that's understandable because you may have a contribution to your ataxia from the gluten sensitivity, and you eliminate that by going gluten-free, but then the genetic condition will then catch up with you because obviously you cannot get rid of the genetic underlying condition. And so over time, the condition will remain progressive, but you may be able to achieve some initial improvement. And I give the choice to the patients. If they're very keen to go on the diet and they come back and say they are much better for a lot of different reasons, then they stay on it.
Well, we are over time. Thank you so much for joining us today, Dr. Hadjivassiliou. We really appreciate your time and your expertise and for teaching us more about gluten ataxia.
Thank you for the invitation.
And for everyone here, thanks for spending the last hour or so with us. We really appreciate your time and hope you learned something new about gluten ataxia today. So wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.
Bye-bye.